Good day, everyone. I'm Sophia Diaz together with Trisha Colmo, Kriselle Lauro, Lorelie Suelto, and Shaina Tomaneng, and we are here to discuss one by one the contents of the sustained effects of ecstasy on the human brain, a prospective neuroimaging study in novel users that is projected in our poster.
To start with the introduction, ecstasy or 3,4-methylene-dioxy or methamphetamine is a substance that alters mood and perception. It is a stimulant and hallucinogen that increases energy, pleasure, and even distorts sensory and time perception. Several studies provided strong indications in which ecstasy can be toxic to serotonin cells. Serotonin is an important neurotransmitter which functions in regulations of mood, sleep, and sexual drive. Some studies examine the acute effects of ecstasy on volunteers without prior ecstasy experience. As a result, the current study established the need to conduct a long-term prospective study to decisively determine ecstasy's detrimental effects on humans by examining the before and after use of 59 novel ecstasy users and also compare them to 58 persistent ecstasy-naive volunteers. The study also considered the following parameters in order to assess ecstasy as a potential neurotoxin. These are the serotonin transporter or SERT and acetyl aspartate or NAA, relative to creatine, myoinositol relative to creatine, apparent diffusion coefficient or ADC, fractional anisotropy or FA, and regional relative cerebral blood volume or RRCBV. Suppose that ecstasy is neurotoxic, the study hypothesized that novel ecstasy users would exhibit a decrease or increase in RRCBV and ADC, increase in choline relative to creatine and myoinositol relative to creatine, and decrease in beta-CIT binding to SERT's FA and the ratio of N-acetyl aspartate to creatine. With that, here's L for the methodology.
In the study's methods, the design and subjects were 59 novel ecstasy users matched individually with a control group of persistent ecstasy naives, so 188 ecstasy naive young adults, 77 males and 111 females, with a relatively high probability of starting to use ecstasy in the near future, were recruited between 2002 and 2004 using a combination of targeted site sampling, the advertisement, and the snowball sampling. So the exclusion of criteria at least 18 to 35 years of age, past ecstasy use, severe physical or mental illness, use of psychotropic medications, pregnancy, intravenous drugs. So the subjects had to abstain from psychoactive substances for at least 2 weeks and from alcohol for at least 1 week before the examinations. So baseline examination, all subjects underwent SPECT and MRI imaging. Next is the assessment of potential confounders. So the demographic variables, education, and substance were measured with the aspects of lifetime ecstasy use, alcohol consumption, tobacco, cannabis, amphetamines, and cocaine uses. So MRI acquisition and processing. Acquisitions, MRIs were performed on a 1.5T scanner, the Signa Horizon LX 9.0, with the use of standard head coils. With the use of MRI, axial proton density, and T2-weighted imaging, the 3-voxel-based HMRS scans, DDI, PWI, and high-resolution T1-weighted 3D imaging were acquired. So post-processing, the scan analysis was analyzed by the LC model or the linear combination model spectra. Image A is the HMRS after analysis by the LC model and representative. B image is the FA. C is the ADC, D is the RRCBV, and E is the binding image transformation to the spatially normalized MRI brain template. So for SPECT acquisition and processing, SPECT imaging was performed with radiogland that binds with non-selectively to SERTs. So SPECT images were acquired for 4 hours after the injection. When stable specific binding uptake to the SERTs is reached, ROI and voxel by voxel analysis were performed. Voxel by voxel analysis was performed with statistical parametric mapping software. So for the statistical analysis part of the study, the substance use variables were log transformed because they were not normally distributed, and future ecstasy use between follow-ups are binary variables since it will be novel ecstasy users versus the persistent ecstasy naives volunteers. So the baseline differences were analyzed with a chi-square test and age, and to test whether ecstasy had an effect on imaging parameters, linear regression analysis were performed. So ROI analysis was done with the use of SPSS 1.5 model, and the SPEC voxel by voxel was analyzed by the spatial extent statistical theory SPM2. So now I shall proceed with study's results.
Figure 2 displays the statistically significant results from beta CAT SPECT, HMRS, DPI, and PWI, which are presented as graphs of areas with significantly different findings between novel ecstasy users and persistent ecstasy naives at follow-up in marked brain regions, the FA thalamus, globus pallidus, and frontoparietal white matter, ADC thalamus, or CBV globus pallidus, and putamen. Novel ecstasy users and persistent ecstasy naives did not significantly differ on any of the neuroimaging parameters and effects of ecstasy based on the brain metabolite ratios. However, the former exhibited a notable difference in the FA neuroimaging in the thalamus, P is equal to 0.025, and frontoparietal white matter, P is equal to 0.009. RRCVV in the globus pallidus, P is equal to 0.022, and putamen, P is equal to 0.029, while a significant increase of Fa in the globus pallidus, P is equal to 0.20, and ADC in the thalamus, P is equal to 0.017, compared to the latter for non-users. Now, we will proceed with the discussion of the said results.
From the acquired data, it was shown that even a low to moderate ecstasy dose of 1 to 80 tablets still has some sustained effects on the brain. Decreased Fa, which reflects the brain's connectivity, and an increased value of the ADC in the thalamus may reflect axonal damage induced by ecstasy, because axonal cell membranes are known to be responsible for most of the restriction of water diffusion. This finding of ecstasy-induced brain pathology in the thalamus corroborates findings from previous studies showing decreased thalamic SERT, or serotonin transporter, in heavy ecstasy users, most probably reflecting damage to terminals of serotonergic axons. Now, from the reduced Fa in the frontoparietal white matter, it was deduced to have reflected abnormal brain maturation, as differences in Fa values between novel ecstasy users and persistent ecstasy naive subjects in this study resulted from a combination of a relative decrease in Fa in novel ecstasy users and an increase in Fa in persistent ecstasy naives during the follow-up period. However, there were some inconsistencies in the amounts of RRCBV, or regional relative cerebral blood volume, and ADC detected in comparison to previous studies, which may be due to the relationship between ecstasy use and brain perfusion in relation to time since last tablet, cumulative dose, and adaptation of serotonergic transporters and receptors. Moreover, no significant differences in substance use, SERT polymorphisms, and imaging parameters were found between future ecstasy users and persistent ecstasy naives at baseline. This is important because it has been repeatedly suggested that observed differences between ecstasy users and non-users in previous studies may have been a pre-existing risk factor for ecstasy use rather than a consequence of this drug.
So, culminating all the data, most of the neuroimaging parameters of neurotoxicity showed no evidence of adverse effects of relatively low dosages of ecstasy on the brain. On the other hand, though, there were significant effects of low-dose ecstasy use on perfusion-rated imaging and diffusion tensor imaging, even after a mean abstinence period of 18.7 weeks. As expected with such low dosages, the effects were rather small, with differences between the groups being less than 5%. A recently published article in almost the same prospective study group, even after lower cumulative dose, showed a small but significant decrease in performance of verbal memory tests in the new ecstasy users compared to the persistent ecstasy naive controls. Although the effects are small and within normal range, it is worrisome that even small amounts of ecstasy seem to have measurable adverse effects. These effects may become more clinically significant with increasing age when the serotonergic function decreases physiologically. Moreover, a recent study with exactly the same imaging parameters showed greater effects in heavy ecstasy users, and thus the effects seem to be a dose-dependent, as previously also suggested by others. And this will be the end of our poster presentation of the sustained effects of ecstasy on the human brain, a prospective neuroimaging study. Thank you for listening, and we hope that you've learned something.